Right, now we will talk about the concepts of desensitization. How the receptors and signaling mechanisms can be desensitized. First we talk about what is the concept of desensitization. Actually, when a specific agonist is continuously stimulating a receptor and signaling mechanism, then with the time receptor and signaling mechanism become less efficient. It becomes less efficient and response to the same concentration of the agonist progressively decreases. Right? Let me give you first example of desensitization from the practical life, romantic life, from marital life. It's very common that before the marriage, men are very super sensitive to the would-be wife. But after the marriage, men start getting desensitized to their wife. For example, if wife is very demanding, right? She is too much demanding and too much interaction. Whenever there's excessive interaction of anything, biological system may undergo desensitization. Right? Now if wife is too much stimulatory, or you can say demanding on the husband, or interacting too much, after some time, to prevent the overstimulation, maybe the person put cotton in his ears so that he doesn't listen much. Right? This is early stage of desensitization, that she is talking but he doesn't listen. He is still in the home but doesn't listen. But if she still keep on having agonist action, then what he will do? He will run out of the home. He will spend more time out of the home. He will spend time where his wife cannot approach and do some agonist activity. Is that right? But if still, whenever he comes back sometimes, she's too much agonist activity, what he will do? Most probably he will commit suicide. This is what exactly happens with the receptor and molecular system. That when receptors are too much stimulated, first they put the cotton plugs. What are cotton plugs? There are no cotton in the cell. The cotton plug for the receptor is phosphorylation, phosphates. When receptors are overstimulated, they get phosphorylated and they become less sensitive to agonist. If still stimulation is too much, the phosphorylated receptor go intracellular. We call it sequestration. They run away from the cell surface to avoid the agonist activity. Right? And if they turn back to the membrane and again there's too much stimulation, then they go inside the cell and get catabolized, committing suicide. Let me explain step by step. Let's suppose that here is a cell and here this cell has adrenergic receptor. Let's suppose this is beta 1 adrenergic receptor, and the same cell, let's suppose, has one more receptor which is called alpha 2 adrenergic receptor. All of you know that beta 1 adrenergic receptor stimulate G stimulatory, alpha component of G stimulatory, and alpha 2 adrenergic receptor stimulate intracellularly, yes, alpha component of G inhibitory. And then, as you happen to be very intelligent, so you must be knowing that if here is your friend adenylyl cyclase, right, here G inhibitory can interact and here G stimulatory can interact, right? So when G stimulatory or alpha stimulatory interact here, this stimulate adenylyl cyclase, right? That will lead to stimulation of adenylyl cyclase activity. But if G inhibitory is activated through alpha 2 adrenergic receptors, then what will happen? That G inhibitory will engage here and G inhibitory will lead to inhibition of adenylyl cyclase activity. And what is the activity of adenylyl cyclase? Adenylyl cyclase is responsible to convert, yes, ATP into cyclic AMP, and cyclic AMP, yes, stimulates protein kinase A. It stimulates protein kinase A. Is that right? Now let's suppose that if we are stimulating beta 1 adrenergic receptor strongly, there are a lot of epinephrine molecules and there is strong stimulation of beta 1 adrenergic receptors. Now when these receptors are stimulated, right, their intracellular component undergo a change, and this alteration in intracellular component of the receptor has two effects. One effect is it will stimulate G stimulatory. Other effect is that it will open a very special area, and with this area there is an enzyme inside. It will activate here. The enzyme is like I don't know how to make a dog. Do you know how to make a dog? Okay, let's suppose this is some dog. I don't know, it's unusual dog, I think. Right. This is barking. B-A-R-K. This is an enzyme present within the cell all the time. All those cells which are beta 1 adrenergic receptors. Right. This enzyme is barking. Why? Because its name is beta adrenergic receptor kinase. What is the name of this enzyme? Yes, beta adrenergic, yes, receptor kinase. It means that it can do kinase activity with beta adrenergic receptor. Right? This enzyme can do kinase activity on beta adrenergic receptor. But when it will do kinase activity? It will do kinase activity to beta adrenergic receptor only when beta adrenergic receptor is loaded with the agonist. Right? So whenever beta adrenergic receptor are loaded with the agonist, they are bound with the agonist, they are bound with, suppose, epinephrine, right? This alters an intracellular domain, and this intracellular part of the molecule becomes available to this doggy enzyme. And this doggy enzyme, which is called beta adrenergic receptor kinase, will act on that point. And what it will do? It will phosphorylate. It will phosphorylate some part of the receptor. And when this part of the receptor is phosphorylated, when this part of the receptor is, yes, what is happening to this part of the receptor? It is getting phosphorylated, then receptor become resistant to the action of the agonist. So what has happened? We can say that receptor has, yes, what has happened here? The receptor has been phosphorylated, and this phosphorylated receptor will undergo further change. When this receptor becomes phosphorylated, then there is another protein present in. This protein will go and bind with the phosphorylated. Okay, I will make this protein like that. Right? This protein will go and bind with phosphorylated receptor. Right? And when this protein will bind with phosphorylated receptor, when this protein has bound the phosphorylated receptor, do you think now G stimulatory can bind here? Attention please, let me review. What happened? Epinephrine came, stimulated the receptor, receptor stimulated the G protein, and of course alpha component of G protein went to stimulate the adenylyl cyclase. Is that clear? Meanwhile, receptor got phosphorylated by the beta adrenergic receptor kinase. When receptor got phosphorylated, then another protein went and bind with the phosphorylated receptor. And when this protein bind with the phosphorylated receptor, can alpha component again bind with this? No. It means presence of this protein will arrest the signaling mechanism. Are you understanding? presence of this protein will arrest the signaling mechanism. So this protein is very appropriately called beta arrestin. Arrestin. What is the name of this protein? This is called beta arrestin, right? And once receptor get phosphorylated by beta adrenergic receptor kinase, and then receptor get loaded with beta arrestin, do you think then agonist may be still binding here, but can signals go forward? No. So it means receptor system is getting desensitized to the presence of agonist. That receptor is having the agonist but not passing the signals forward. So what will happen to the same dose and same concentration of the agonist, the response of the cell and tissue will be less. So we'll say receptors is desensitized, or we can say cells are desensitized, or we can say tissue is desensitized, or even we can say patient is desensitized to this particular drug. Am I right? This type of receptor phosphorylation and binding of the arrestin with the receptor produces desensitization within few seconds. Is that right? So we call it rapid desensitization. This mechanism is rapid, yes please, rapid desensitization. Is that right? If walking, again go back to that example, when wife is talking too much to the husband, he is worried about his office matter but she is more worried about her shopping, and she is talking and talking and talking, what will happen? First, husband will get phosphorylated, and maybe some other woman come and bind there, and even arrestin activity may occur. So be aware, both parties, right? It's dangerous for both. Am I right? And this type of desensitization can occur rapidly. Is that right? Now, but remember, if we remove the agonist for short time, wife become less demanding and less about the shopping, right? He will turn back, most probably. If she in time, she withdraws. Same is true that if this agonist for a short time is kept away, right? The arrestin will go away, dephosphorylation will occur, and receptor will become what? Again, sensitive. Am I right? This is one example. Then there is another type of desensitization. Let me show you here. This type of desensitization is called, yes, homologous. This is an example of homologous desensitization. Desensitization. What is meant by the homologous desensitization? Homologous desensitization means that agonists bind with one receptor and the same receptor becomes desensitized. Is that right? Now you imagine another thing. Listen carefully first to the example. There are few couples living together in an apartment building, and one wife is very hard on her husband, right? Always demanding for shopping, I want this, and shouting, and everyone is listening. So not only husband of that wife will become desensitized, the risk is that other husbands in the building may also get desensitized. That wives are like that, and they may not listen to their wife, other husbands may not listen to their genuine demands of their wife. That is called heterologous desensitization. Are you understanding me? Really? Right? So, again, if my wife is shouting to me a lot, and if I get desensitized by phosphorylation and arrestin, what is this? Homologous desensitization. But if other husbands see what's happening with me, and they learn something, and they also become desensitized to their wife, it means too much agonist activity on me lead to my homologous desensitization, and other receptors, other husbands, heterologous desensitization. Let me tell you the exact molecular mechanism of heterologous desensitization. Let's suppose, this receptor system is giving signal, then what will happen? Adenylyl cyclase will be activated, right? And then protein kinase A will be stimulated. Protein kinase A does many functions. You remember it does phosphorylation of target proteins, and target proteins will undergo phosphorylation. Phosphorylation of target proteins. But you will be surprised to know that protein kinase A may also do phosphorylation, may do the phosphorylation of receptor which stimulated, as well as it may do phosphorylation of some other receptors which are working through the same signaling mechanism. You are getting it? And I told you when receptors are getting phosphorylated, they are becoming less efficient. So what was happening? That this receptor was having too much agonist activity, stimulating its G-stimulatory, stimulating adenylyl cyclase, getting more cyclic AMP, increased activity of protein kinase A. And this protein kinase A will phosphorylate many proteins, including some receptors which are acting through the same molecular mechanisms. Is that right? So if this receptor, which is alpha-2 adrenergic receptor, that is phosphorylated, it means that sensitivity of this receptor and response of this receptor mechanism to the agonist will be also reduced. This type of receptor desensitization is called heterologous desensitization. What is this called? Heterologous desensitization of receptors. Is that right? Remember, in homologous desensitization, phosphorylation is followed by attachment of arrestin. But in heterologous desensitization, there is simple phosphorylation. But that is also enough to bring some decreased response of the receptor mechanism. Am I right? Any question up to this? So this is a rapid type of desensitization. Then there can be relatively slow mechanisms of desensitization. As I told you, don't forget. Let's go to our basic examples of desensitization. Wife demanding husband too much. What will happen? That husband does not listen, and even he gets some arrestin, some beautiful other lady, right? What is that? Rapid homologous desensitization. But if that husband is suffering, listening to wife, doing a lot of shopping and suffering financially, right? Too much agonist activity, then other husband take a lesson from him, and they don't listen to their wives, unfortunately. What is that? Heterologous desensitization. But if that wife is really very persistent, she believes in perseverance, so then what will happen? She disturbs him too much agonist activity. What will happen? He will run away from home. He will run away from home, and he would like to stay away from his wife. He will like to stay at places where his wife cannot come with the demands and agonist activity. Same thing happens with the receptors also. If they are really too much stimulated, they decide to run away from the cell membrane to inside and hide into cell, like the poor husband, right? So what really happens that, let me put simple diagrams of these receptors. What are these receptors? Suppose adrenergic receptors. All these receptors are strongly stimulated. When these are strongly stimulated, I told you they will get phosphorylated. If receptors remain phosphorylated repeatedly, then receptors bind with some special intracellular proteins. These intracellular proteins are called calatherins. What are the name of this protein? These proteins are called calatherins. These phosphorylated receptor, especially with arrestin, they bind with the calatherins, and calatherins are special proteins which pull the membrane inside. They pull the membrane inside. So this piece of the cell membrane loaded with the receptor will go in, right? And receptors are still present over there. You are understanding? And they are still having what? Phosphorylation, you remember? And they are also having their, okay, I will change the color. What was this? Calatherine. So calatherine, we call it, this calatherine coated part of the membrane pulls in. So this is a vesicle is being formed. After a little time, this will go inside the cell. Now inside the cell, this was calithrin coated pit. Now it has become intracellular vesicle. Receptors have escaped from the cell membrane or cell surface to the intracellular area. Is that right? And they are still phosphorylated. What are they? Phosphorylated and calithrin coated. Then what happened? When receptors have gone in, can epinephrine stimulate these receptors which have gone intracellular? No, because epinephrine cannot go into cell. Because epinephrine molecule is highly polar molecule. It is highly charged molecule. So it cannot go in. So in a way, when receptors have gone in the intracellular vesicle inside the cytoplasm, they are not available to the agonist. So tissue response to that agonist will reduce. Right? These receptors remain in for short time. Right? As husband ran away from house. And when he will go away after, for example, for 2-3 days, and then he will remember, he will forget his anger, and he will remember, no, my wife is good, there was a time I used to love, still I can try something. And maybe as it happened, he will come back. Same happens with these receptors. What happened? That they remain inside sequestered. We say receptors have been sequestered. This process is called sequestration. What is this mechanism called? Sequestration. So we say that not over second, over minutes, many receptors are internalized by the cell and they are sequestered by the cell, and number of receptors transiently available on the membrane are less. But when receptors go in, like that poor husband, right, they again become activated. How? That there are enzymes here which are called phosphatases. They will dephosphorylate it, and they are now again ready to undergo action. Right? And after that, when they become dephosphorylated and they become again fully sensitized, what will happen? You know what will happen? What happened to that husband? He came back. Right? So this vesicle will come to the surface. Right? And receptors are again available on the surface. So this was a transient, what was this? A transient desensitization. So listen again. Previously we talked about that receptor was phosphorylated. It was available on the surface but not responding well. That is called rapid desensitization by phosphorylation. In second example we took that when receptors are really stimulated too much, eventually they undergo process of sequestration. Right? They go inside the cell and they hide it from the stimulating agent. There they get dephosphorylated and again develop their full sensitivity to the membrane and again recycle on the surface. Is that right? So that they have come back to the home and again that wife is there, but unfortunately she has not learned much. She come back with all her demands. Look at her. She's very happy that stupid man is back. Right? And now again too much stimulation. It means this group of receptor will again and again cycle in. Look, it will get phosphorylated, cycle in, sequester, dephosphorylated, and then again re-express. But every time when it is treated with too much stimulation over long time, over hours, oh my God, you know what will happen to that man? Repeatedly come back, suffer too much, again goes, get emotional energy, come back, again suffer. Eventually he will decide to commit suicide. So same will happen to this receptor business, that eventually with repeated sequestration cycles, they become so heavily phosphorylated, they cannot be dephosphorylated, and then these vesicles take a different pathway. They don't express on the surface again. Man does not come back home. What happened to this man now? Not man, I think we were talking about receptors. These receptors, which were initially transiently overstimulated and they were phosphorylated and sequestered, now they will fuse with, what is this? This is endosomes. Lysosomes. Every cell has lysosome with dangerous enzymes. These are lysosomes with dangerous enzyme. Eventually, these endocytic vesicles loaded with the receptors will fuse with the lysosomes, and very dangerous lysosomal enzymes will attack the receptors, and then receptors will be catabolized. And these receptors which are catabolized, of course, they will not cycle back. And what will happen? The receptor concentration or receptor density on the cell membrane will be reduced for long time. Is that right? This is called down regulation of receptor. This is called down regulation of receptor. Is that right? Now, down regulation of receptor occurs in many ways. One way is the receptors which are already overstimulated, they are internalized and eventually they bind with the lysosomal, their endosome bind with the lysosomes, and then receptors are destroyed. So we can say there is increased catabolism of receptor. What is there? There is increased catabolism of receptors. Receptors have committed suicide. And listen, if many receptors are committed suicide, okay, listen, in your town, if many husbands are committing suicides, do you think young boys will love to marry? It's a good example or a bad example? Marriage rate will go down. This is exactly what happened in the cell also. That when many receptors are committing suicide, they are internalizing and they are undergoing catabolism, the signals go to the nucleus. And in the nucleus, if this is the nucleus, this is the DNA. You know how receptors are made? From the DNA we make? Yes. From the DNA we transcribe RNA. And then RNA come out, bind with the ribosomes, and then peptide chain is made, and this peptide chain convert into receptor. Is that right? This is the normal mechanism. DNA to RNA to DNA transcribed into RNA, and RNA is translated into peptide, and this peptide will appear on the surface as receptor. Normally, small percentage of receptors are going under catabolism, and small percentage of new receptors are continuously being made. This is normal physiological receptor turnover mechanism. But when receptors are too much stimulated, the rate of catabolism becomes fast. At the same time, signals go to this all synthetic process, and this synthetic process also get inhibited. Those cells which are overstimulated by an agonist, then messenger RNA, for the formation of those receptors which are overstimulated, that messenger RNA undergoes less concentration. So it means synthetic processes are also down regulated. Is that right? Am I clear? So what really happens, that eventually synthesis of receptors become less, catabolism of receptor becomes more, and number of receptor on the cell membrane become permanently low. Is that right? Whole this process is called, yes please, what is this called? Down regulation of receptors. Down regulation of receptor as means there is permanent, yes, decrease in receptor number by two mechanisms: increased catabolism of receptor plus there is decreased synthesis of receptor. Am I really clear to you? Is that right? So all this is called receptor signaling mechanisms desensitization. Is that right? Again, I will just make it briefly, that when agonists come and overstimulate a receptor, first of all, receptor will undergo phosphorylation and undergo rapid desensitization. Receptor stays there but does not listen. Right? This is the mechanism. Phosphorylation is two types. There is homologous desensitization and there is heterologous desensitization. In homologous desensitization, the receptor which is loaded with the agonist, this receptor exposes the domain, intracellular domain, to be acted upon by the beta adrenergic receptor kinases. This beta adrenergic receptor kinases is a whole family of enzymes, kinases, which are called G protein coupled receptor kinases. This dog belongs to a big family of dogs. All those dogs actually phosphorylate the G protein coupled receptor. So beta adrenergic receptor kinase is the one member of G protein coupled receptors kinases, right? When receptor is loaded, this will phosphorylate that, and then with the phosphorylated receptor, arrestin will bind. After that, when receptor has phosphorylated and bound with the arrestin intracellularly, then G proteins cannot bind with this and receptor cannot give signal, right? This is homologous desensitization. Another example is of desensitization when receptor has given signal and activated adenylyl cyclase, cyclic AMP has gone up, protein kinase A is stimulated. Stimulated protein kinase A will do many target function, but it may also phosphorylate the same receptor which produce the whole activity, plus it may phosphorylate some other receptors which are also acting through the common, what is this, effector system. And when other receptors are phosphorylated and they become desensitized to their agonist, we say there is heterologous desensitization. Then I told you, if there's too much stimulation, now receptor have to run away, at least transiently. That is an example where phosphorylated receptors, especially with arrestin, bind the clathrins and they make, what is this, endocytic vesicle, and transiently receptor disappear from the membrane to the internal environment and become unavailable to the agonist. But initially when the vesicles are formed, they are internalized, there is sequestration of the receptors, phosphatases will dephosphorylate the receptor, and like a stupid husband, they will come back to the membrane. When they will come back to the membrane, if wife is not intelligent, she will again push more and more agonist activity. If repeatedly sequestration, internalization and recycling, these cycles go many times, eventually a change comes and endocytic vesicle with receptor start fusing with the lysosomal granules, and lysosomal enzyme destroy the receptors, and then receptors number become permanently less. Meanwhile, signals go to the nucleus, the receptors are too much stimulated, we don't need so much receptors there, and what really happens that messenger RNA which is involved in the synthesis of this receptor, that is also reduced. So the net result is over prolonged stimulation that increase catabolism of receptors and decrease synthesis of receptor, and receptor number on the cell membrane become permanently low, and we say that there is down regulation of receptor. So there are three types of desensitization, right? Rapid desensitization by phosphorylation, right? Then there is transient disappearance of receptors, sequestration, and permanently receptors are reduced, yes, down regulation. Am I clear? For sure. Now let me tell you another thing. Have you heard of upregulation of receptors? Have you heard of it? You know the concept, or should I explain? That when a group of receptors are not stimulated on the membrane, they become more sensitive to the agonist. Let me tell you the practical example. For example, a person's wife is very good, but she goes to her parents for 2-3 months and he misses her too much. Then what will happen? He will become less sensitive to her or more sensitive to her? He will become so sensitive, even again he will talk on the telephone very romantic things, but she should stay for enough time there. You are understanding? That is upregulation of husband's emotions, right? But remember, a little distance increases love, but too much distance decreases it. Someone else may come, right? Now let's come back. I don't know, you laugh at that. This is a very important thing, more important than medical science. Anyway, let's come back. So what I'm talking about, receptors can be upregulated also. Let me give you an example here. Let's suppose this is beta 1 receptors, right? Okay, I'll give the example here. Let's suppose this is a cell from the heart. These are beta 1 adrenergic receptor. You are giving the person propranolol for many months and years. For example, I'm a person with hypertension and you give me propranolol, beta blocker, and there's a very good response and my blood pressure is controlled. So you keep me on propranolol for a long time. Meanwhile, if I get some, what is that? Well controlled. If here is propranolol and propranolol is blocking the receptors. If these receptors are blocked, do you think epinephrine can stimulate these receptors? No, epinephrine cannot stimulate these receptors. So there is understimulation of receptor. When receptors are understimulated, none of them will be phosphorylated. None of them will be sequestered. All of them will be on the surface. And nucleus will be getting message. Whenever receptors are activated, a message goes up to the nucleus. That receptor and signaling mechanism is working. But when receptors are blocked by agonist, nucleus loses those signals. So nucleus assumes, the genetic machinery assumes that if there is no signal coming, probably receptors are not there. So they will start expressing receptor associated gene more, and receptor synthesis will increase. Remember, if you overstimulate the receptor, receptor genes underexpress. If you do not stimulate the receptors, genetic operators think probably why there is no signal coming, probably there are less receptor. So genetic operators start synthesizing more receptors. So what will happen? What will happen? Synthesis of receptor will be more, and receptor will become more and more and more and more and more. Membrane become overloaded with the receptors, and you keep on giving propranolol and keep on them blocking them. Is that right? Now receptors are upregulated, but you are keeping them blocked with propranolol. It is a very big clinical significance. If I'm hypertensive patient, you are giving propranolol for a long time, and on my heart beta 1 receptors have been blocked for a long time, then receptors are upregulated. Their concentration on the SA node, AV node and other myocardial tissue become very high. And if due to some doctor who doesn't know these things, one day I go to doctor and I say my hypertension is not being controlled by beta receptor. He checked my hypertension, and with the time, over 5 years, my hypertension is no more controlled by propranolol. And that doctor decided to change my drug, and he abruptly stopped my beta blocker, and all receptors become available for endogenous epinephrine. Heart is super driven by the endogenous epinephrine. Heart rate goes up, contractility goes up, palpitations are there, and if I have some coronary artery disease due to hypertension, myocardial infarction may precipitate, only because doctor did not know that if someone has taken beta blockers for a long time and his receptors will be upregulated, in such cases propranolol should not be stopped abruptly, it should be gradually tapered off. Is that right? So as doctors, even though I was just talking about adrenergic receptor, but in whole pharmacology you must be clear about the concept of receptors desensitization, sequestration and downregulation, as well as you must be very clear about concepts of receptor upregulation.